So DNA sequencing is based on our understanding of replication, that is to say, what happens during replication. It is also based on the design of some anti-cancer drugs, on some chemotherapeutic drugs used to battle cancer. So let's talk about that first. Among the various chemotherapies that are intended to attack the ability of cancer cells to grow and reproduce and replicate their DNA, these are things that the cancer cells do more so than other cells. So if you target replication of cancer cells with some kind of chemotherapeutic agent, you can slow down the growth of tumors. And so the idea here is to look at this molecule and see why it is a cancer therapeutic agent. First of all, if you look at it closely and you remember some structures, this is really a nucleotide, isn't it? Well, there's something peculiar about this nucleotide. First of all, it is a deoxynucleotide because it is missing the oxygen on the number 2 carbon. But if you look closely, the number 3 carbon is also missing an oxygen. This is called then a dideoxynucleotide. It is missing that number 3 oxygen, which means that while this nucleotide can be added to a growing chain of DNA because its phosphate end is perfectly okay, it has a triphosphate, it's perfectly good 5' end, if you want to call it that, of the sugar. In other words, the number 5 carbon has the phosphates on them, but it doesn't have the oxygen on the number 3 carbon, the OH on the number 3 carbon, which would be the site of additional nucleotides, of the next nucleotide. So if this gets incorporated into a growing strand of DNA at its 3' end, no nucleotides can be added. So that made it a likely therapeutic agent to fight cancer. Incidentally, it blocks replication in all cells that are dividing, which is why this anti-cancer drug, and in fact many others that have broad effects that are directed at dividing cells, will also attack not so rapidly dividing cells like your stem cells and other cells that simply have to divide in order for you to survive. And the result is, of course, all these pretty horrendous side effects that people taking cancer therapies have to endure. But they do work because they preferentially attack cancer cells, which are growing more rapidly than other cells. This drug was first an anti-cancer drug, but then was co-opted to do DNA sequencing. So let's look at, in principle, what's going on here. This process of sequencing is called dideoxynucleotide DNA sequencing. So we have 4 tubes here. Each of them contains essentially the same stuff. The only difference between them is which of 4 dideoxynucleotides they contain. One is going to contain dideoxy-ATP, one is going to contain dideoxy-GTP, the third dideoxy-CTP, and the fourth dideoxy-TTP, right? Adenosine triphosphate, guanine triphosphate, or guanosine triphosphate, cytosine triphosphate, thymidine triphosphate. They look like the precursors to DNA, but they are the dideoxys, so if they get incorporated into chains, no further growth will occur at that point. So what are the other things in these tubes? Well, they contain everything else you need to do cell-free replication, to do DNA synthesis in a test tube. So they will contain DNA polymerase. They will contain the 4 natural deoxytriphosphate, deoxythymidine triphosphate, deoxycytosine triphosphate, and so on. One of them, usually deoxyadenosine triphosphate or deoxy-ATP, is radioactive. In addition, you put into this tube the DNA that is to be sequenced. So it's the template DNA that's going to be sequenced. Dideoxy sequencing is really a replication-based sequence. Well, what else do you need to make this happen? Well, you're going to need a primer. That is usually a synthetic oligonucleotide that is based on the sequence of the gene that you wish to sequence. So it's a sequence you have to know in advance. So you need a primer because DNA replication requires a primer. To summarize, you need all the components necessary to do DNA sequencing based on the known activities of polymerase, the known precursor requirements. One is radioactive, so you can follow it. You need template that's going to be sequenced, and the primer, so that the DNA polymerase can add to the primer in order to do the sequence. And you need to add these dideoxys, 1 each. That's what makes each tube different. So this is what's going to happen as the DNA is actually synthesized in each tube. So let's imagine we have lots of template. Those are the blue strands. We have annealed an oligonucleotide primer to which we're now going to be adding new nucleotide. The sample on the left is in the tube that contains dideoxy-A. The tube next to that contains dideoxy-C, and so on. So what do you expect is going to happen, say, on the tube on the left? As DNA is synthesized, if you have put the right proportions of ingredients into these tubes, every now and then a dideoxy-A is going to be added to the end of a growing chain, and at that point, that chain cannot grow any further. So the idea is that we will have made, on a random basis, several different length new molecules of DNA, each one extended until the incorporation of one of these dideoxy-A molecules. At that point, they can't grow anymore. So we have a distribution of different fragments within a dideoxy-A opposite a T in the template. Let's take a look at the C. So, and in fact, we can look at all of them at once. Dideoxy-C fragments are terminated wherever there was a G in the template, and if you did it just right, you get a fair distribution of all the different possible fragments that would terminate where there is a G in the template. If you use dideoxy-G, you will be terminating fragments opposite where there is a C in the template. And when this is done, you can get fragments up to 700 or 800 bases long, representing every possible G or every possible A or every possible C or T in the template with a cognate or complementary fragment terminated with one of the dideoxys. So now you have all these different size fragments. So what you do is you run a gel of the material from each of these tubes. These are now radioactive, remember, because into each of these fragments a certain amount of deoxy-ATP, the normal nucleotide, has been incorporated, so there are going to be radioactive A's in these sequences. And so you can run the gel and then place a piece of X-ray film on top of the gel, expose it, develop it, and you'll get something that looks like this. It's a piece of X-ray film with lots of bands. What's really miraculous, if you want to think about it that way, I used to say in classes, this is about as miraculous as a 747 getting off the ground. This is a length separation, a size separation. Each band is separated from its nearest neighbor by the amount of DNA in one nucleotide. And since you can get fragments on a system like this up to 300 bases long, what we're really saying is when you look at a real autoradiograph, you can see pieces of DNA that differ in length by one nucleotide. In the case of a 300 base long fragment that you're trying to sequence, that means you're looking at distances that are 1/300th of the entire length of the sequence that you're looking for. Now, how do you read a sequence? Well, you actually read from the bottom up. So the sequence, if you look at it on the gel, before I show it to you, would read something like C, G, A, T, G, G, etc. So you read in order of the ladder. The first base is a C, that's the lowest one down. The next one up is a G. The next one up after that is an A, and so on. It's hard to do that without a pointer, but I think you get the idea. Well, this process is what I used in my lab to do a lot of sequencing in the early days. The process became automated, so we don't need to go into great detail about how this automated sequencing is done, but basically it uses dideoxynucleotides that are fluorescent, and instead of running 4 different tubes, you run it all in one tube, and you put all the dideoxys in together with everything you need to make DNA, except that each dideoxy fluoresces a different color, and we have a big machine that detects the colors as they come off of a gel. It's still a gel. The neat thing about this system was where we would do 300 bases at a time, manually doing actual X-ray film analysis. The automated sequencing could give us sequences of up to 500 and 600 bases, even 700 bases on a good night when the moon is full. And now there are technologies that can give us even more. The take-home from this, of course, is that you can get a lot more sequence a lot faster. And it was with the help of machines that projects like the human genome could be accomplished. Originally, if I remember right, the Human Genome Project, when it began, was predicted to take 15 years or so. It ended up being done in, I forget exactly, but 5 or 6 years. Because advances in the technologies for doing this kind of sequencing in an automated fashion. And so what you're looking at is a chromatograph representing the 4 different fluorescent dideoxys that are incorporated into the different fragments that were made, each of a different length, terminated opposite the appropriate complementary base in the template. So that's DNA sequencing.
And that brings us to the end of this presentation.